All right, so we're going to be looking at the different methods for diagnosing monogenic diseases. So these are things like single nucleotide variants, or SNVs, where you had some kind of substitution at a place or an insertion or deletion at a place, and it affects a gene in your DNA, and this leads to a defective protein that leads to some kind of disease. And so there's many different ways of doing this. The first is known as polymerase chain reaction, or PCR. And a key thing to make note of with a lot of these diagnostic tests is that with almost all of them, except the last one we'll talk about, we already know the bad sequence of gene, or we know what the wild type sequence should be. So like we'll know exactly what the DNA should look like, so that when, if we took a swab from a patient's cheek or a blood sample that had DNA in it, and we noted that, it didn't have what we're looking for, or it had what we're looking for in it, we already know what we're looking for is the key point here. That's how we're going to identify it, and that's how we're using PCR. So, the COVID tests that were out, people were able to do PCR tests to literally check for exactly the sequence of the SARS-CoV-2 virus, because we literally had already sequenced that virus. So we knew exactly what we were looking for, which is how we used PCR in the first place. And so the reason why you need to know it a priori is that you will have primers that are very specific for a region of interest. So if we looked at COVID, for instance, we had primers that would anneal specifically to the DNA sequence for the coronavirus, and because of that, we would be able to see if we took your random DNA and the primers were able to anneal. If the primers were able to anneal, then the polymerase chain reaction would end up creating a bunch of genetic material when we run it, and that would mean that you did have it, because we did detect the coronavirus genetic material in your cells. So that's what PCR does. And so we're able to identify if you've got some kind of pathogen genetic material present, like HIV or the SARS-CoV-2 virus. We're also able to detect if you have some kind of known mutation. So we can make a primer specific for that mutation that would tell us exactly whether or not you have it. And these could be things like insertion deletion mutations that we've already sequenced in the past, or stuff like that. So that's what PCR does. Now, there's another thing called PCR-RFLP, or RFLP, as I think of it in my head. So what it stands for is restriction fragment length polymorphism. And so the key thing to take away from this is know that with DNA, we've got restriction fragments, and what these are places where you can have restriction endonucleases, which are proteins that will cut up DNA. They'll cut it in places where they hadn't cut it previously, or the mutation could act the other way and cause DNA to not have these specific cut sites, because they just got changed a little bit, or just enough so that the enzymes can no longer work. And so basically what will happen is, if you had a mutation that was coincidentally in a place where you would have a restriction enzyme working, then that will change the number of fragments that we would expect to see once we take your DNA and put it through a control experiment where we know exactly how many fragments to expect out of it. So that's basically what we're doing. I want to make note here that this is a very specific type of mutation, and we already know it beforehand. So we know that it will affect the restriction fragment count, and so this is how this method's working. So if we do or do not see a band where we expect to see it, that's indicative of someone having a genetic disease, a monogenic disease. The next one we'll be looking at is called ARMS-PCR. This is called amplification refractory mutation system. And so again, this is a little bit less specific than the RFLP PCR, because this can be any point mutation anywhere, but we are relying on it because what we would have already known beforehand is that, let's say that instead of T at some nucleotide position you had G, we would make a primer so that the 3' end of the primer, so the very last nucleotide of that primer, would not anneal onto that nucleotide where that point mutation happened. And because of that, the DNA polymerase is not going to be able to actually build the complementary strand, or the nascent strand. And so if we basically take patient's genetic material and we anneal these nucleotides with that special engineered 3' end that will only anneal to the wild type protein, or we could also do a negative and look at only the ones that will anneal to that specific mutation that we're looking for. If we see amplification or we don't see amplification, when we do a gel and we look at the bands, that will tell us if that mutation is or is not present. So that's how ARMS-PCR works. A little bit more general than RFLP, but still pretty specific in that we need to know exactly what we're looking for beforehand. And then we've got another one which is called ASO, or Allele-Specific Oligonucleotide. And what we do here is we basically will know beforehand the sequence we're looking for, and we're going to make a bunch of single-stranded DNA. So we'll take double-stranded DNA, make it a single-stranded DNA, and then because we'll know the sequence that we're looking for, we can engineer oligonucleotides to have affinity and to hybridize with specific mutations that we're looking for. And so this is another way that's basically very, very similar to what PCR is doing, because PCR, we've engineered those primers to only stick to specific things that we wanted them to, but in this case we're having this stick onto, we have things on surfaces of chips that will basically hybridize to DNA. And so if we see a lot of hybridization at a particular oligonucleotide place, it will tell us that someone does or does not have a mutation. It depends on the test. But why have this instead of traditional PCR is that basically we have way higher throughput, because you can have a chip with a bunch of oligonucleotides on it, and you can run things called multiplexes where you're basically not just screening for one particular mutation, but you can be screening for hundreds or thousands of mutations if you wanted to. So much higher throughput than PCR, and so it's basically a higher throughput technique that people would use to get results faster and to also check for more things instead of having to do a bunch of one-offs. The next one we're going to be looking at is a technique called Southern blotting. Southern blotting, again, is pretty specific. We already know what we're looking for before we even run this test. The goal here is to visualize the DNA. We're basically trying to see if a particular gene or a variant of a gene is present in that DNA. We're going to, just like with RFLP, those restriction fragment experiments, we're going to chop up that DNA with nucleases. We're going to run a gel. We're going to separate the DNA based on its size and charge, and then we'll use radio labels to hybridize against a target. And so if someone had a triple repeat expansion, so it's like you had a triplet that just kept being multiplied n number of times within DNA, that's a common use case for Southern blotting, because it will help us see it visually. If you do have a region of DNA that just got way more nucleotides in it than we're expecting. So that's the key thing for those types of detections with those mutations. And then finally, what we have are Sanger sequencing. And so Sanger sequencing makes use of these things called dideoxynucleic acids or nucleotides. And so the reason it's dideoxy means that the nucleotide does not have a hydroxy at the 3' carbon. And because of that, when you incorporate a ddNTP, like ddATP, onto your nascent strand, there's no 3' OH to build the subsequent nucleotide onto, so your replication will immediately stop and you get this fragment. And so when you get this fragment, this is going to cause, it'll pop off, and then after you've done this a sufficient number of times, so it's not just going to be ddNTPs, you also have normal nucleotides present. But basically, the end result of this, you're going to end up with like a soup of all these radio-labeled fragments of DNA, and then when you pull your DNA through this thing, you will get different bands at different wells, and from that, you can figure out what is the sequence of DNA that you're looking for. So in this case, if we just zoom in here, you can see how we've got the lowest band right here, which is technically the smallest because it was able to go the farthest. That means that our first nucleotide in this DNA that we're sequencing is going to be T. The next carrier is going to be in well A, because that was the next smallest fragment that pulled farthest through our gel. And then basically you can continue up this chain like so. So that's what gets out of Sanger sequencing. And so now we're going to look a little bit at the summary and basically the considerations that you should make when you're doing this stuff. So out of every single test that we looked at, PCR-RFLP, the restriction of nucleus one, is the most specific, because we're we're basically assuming that the point mutation will be at a restriction endonuclease site. And so if it isn't, then we're not going to see anything. So basically, if we know for sure that we are going to be changing the number of restriction sites in the genetic material, then we can be confident that our PCR-RFLP would work. So this is like, just in terms of how I'm ordering this in my mind, the most specific thing. ARMS-PCR is the next most specific test because we're able to focus in on point mutations, but we have to know exactly where that point mutation is. But at least we're not counting on point mutations that are also point mutations within a restriction endonuclease cutting site. So it's a slightly larger subset, which is the reason why it's a bit more general than PCR-RFLP. And then the next thing up this list of like what is the more general or less specific are the allele-specific tests. So that was the oligonucleotide test. And so it's on the same level as PCR. We're able to, if we know what the mutation is beforehand, or if we know what the genetic materials that we're looking for beforehand, like the coronavirus sequence, we're able to detect these things. So these can be not just point mutations, but insertion deletions or little repeats. As long as we know exactly what we're looking for, we can use them. So a little bit more general than ARMS-PCR and even more general than PCR-RFLP. But the key thing to take away is, like, you know, if you're running this in a hospital, you're going to want something that's way higher throughput. That's the reason why ASO is the much better option here, because you can just run way more tests than you could doing PCR, because PCR is very, very specific for like that one test. And then the gold standard is Sanger sequencing, but it's the most expensive and the most time consuming. So like if a patient presented to you with an unknown disease and you had absolutely no idea what's going on, ordering a Sanger sequencing test will tell you their exact sequence. But again, very expensive, very time consuming, and usually we're only looking at a very small region. So you're gonna have to have a pretty good idea of like what you're looking for already, but they will tell you the sequence of DNA in a region of interest. And so you will be 100% sure if someone does or does not have that genetic disease. Like that is the most specific thing that like, you cannot have any higher confidence than you would with other tests. And then in addition to that, we're also able to check for novel mutations. So everything other than Sanger sequencing here required us to already know exactly what we were looking for. Like we know what the coronavirus genome is, but with Sanger sequencing, like we don't need to know. Like once we capture what we know is the genetic material of, like if you had the HIV RNA or cDNA inside of a patient's cell and you're able to extract that, you don't need to know what the sequence is just yet. You can put it through Sanger sequencing and then you would know what that sequence is. So then, you know, that's how you would begin to create tests for it to screen for that. So that is the key thing to take away from this. And so based on, you know, the type of genetic disease that you suspect someone might have, these are the types of tests to look at. And so I hope this helps, and thank you all for watching.